Hello, this is talk number 31 in the series from the 7th edition of Principles of Biochemistry, written by Albert Lehninger. Uh, Today we're going to kind of put a wide net over much of the material and pathways we've discussed in an effort to integrate some of what we've covered previously, and then have a discussion on obesity. First, we'll discuss the fate of sugars in the organism. Again, as a point of overview, you have the mitochondrion, you have the hepatocyte, you have the blood stream right here. And looking at this in sequence, we have glucose 6-phosphate, which can be converted into glucose and go into the bloodstream. This is in response to energy needs. Alternatively, it can be stored as liver glycogen, or it can be converted through glycolysis into pyruvate. Depending on the immediate energy needs, whether you have an anaerobic pathway which culminates in lactate, or if you go in the aerobic pathway with transport and dehydrogenation with thiamine pyrophosphate and the other cofactors to get acetyl-CoA, and then entering the Krebs citric acid cycle, where electrons go into oxidative phosphorylation and you obtain ATP molecules from that with extrusion of CO2 on the other end. Alternatively, acetyl-CoA can be synthesized into fatty acids or cholesterol as necessary, and the fatty acids can then be further metabolized into storage forms as triacylglycerols or phospholipids used in neurotransmission and overall receptor signaling. For amino acids, we have a slightly different pathway. Again, you've got a situation of amino acids which can be converted into hepatic proteins. Other proteins can be excreted as plasma proteins, or free amino acids going into blood, where they can be converted later into tissue proteins. These amino acids can additionally be converted through the pathways we've just covered into nucleotides, hormones, and porphyrins in the hepatocyte. The breakdown product of amino acids forms ammonia, which can be converted through the urea cycle into urea. Additionally, you can get alanine from muscle as a breakdown product, getting ammonia from that conversion to pyruvate if there's an energy need, with a gluconeogenesis to glucose to form blood glucose and put it into storage form in muscle. Alternatively, blood glucose can be converted if you have pyruvate going into the Krebs carboxylic acid cycle. Again, you can get the same production of ATP, or if necessary, you have intermediates of the citric acid cycle that can be converted to glucose, and this can be utilized in any format that the body deems necessary. For steroid hormones, we have an analogous situation where fatty acids can be synthesized up to liver lipids, or they can be used as beta oxidation under conditions of consistent exercise or energy utilization, forming NADH. These fatty acids can also be converted to acetyl-CoA as 2-carbon breakdown products and go through the citric acid cycle. Under conditions of starvation, you can have ketone bodies in the blood. You can also have free fatty acids in blood and plasma lipoproteins that are converted in the hepatocyte in a sequential VLDL to LDL to HDL form. Acetyl-CoA can also be synthesized up to cholesterol via intermediates, HMD, squalene. These can be converted to bile salts used in intestinal absorption, or they can be modified into steroid hormones in blood. For adipose tissue, we talk about brown fat and yellow fat. Brown fat has more mitochondria. It is literally brown and has a different look under the microscope. This is active in thermogenesis, so you can get heat cycles from this. This predominates in fetus and newborn. Yellow fat is seen in the adult form, and this is triacylglycerol storage, and this is directed primarily to energy conversion. There are fast twitch muscles and slow twitch muscles. The slow twitch muscles are red muscle. This uses oxidative phosphorylation slowly. It is resistant to fatigue, and a higher amount of mitochondria are present in the red muscle. For fast twitch white muscle, this uses oxidative phosphorylation quickly, also fatigues more quickly. There are fewer mitochondria, and you get lactate formation and a rapid onset of energy utilization and pain with the lactate. Epinephrine stimulates these responses. The heart muscle is continuously active and predominantly utilizes free fatty acids, oxidative phosphorylation, and is exquisitely sensitive to oxygen loss. The brain has obligate utilization of glucose. Astrocytes can use fatty acids, but there's no glucose storage. All this is transported by the blood, and you can use ketone bodies in starvation or fasting. In blood, you can utilize glucose. The water is the greatest blood component by weight. Then erythrocytes and the ions present in blood maintain a narrow range of reference interval. There is hormonal regulation of fuel metabolism. Insulin and glucagon have opposing effects on glucose uptake, glycogen, fatty acid synthesis, and glycolysis. Epinephrine and cortisol affect the short and long-term energy utilization. When you consume a high glucose meal, the GLUT transport-2 transporters carry glucose into pancreatic beta cells. Glucose then enters glycolysis. There's an ATP increase, and this closes the ATP-gated potassium channels. The subsequent loss of potassium efflux creates membrane depolarization, and then calcium enters, and the intracellular calcium that enters triggers insulin release, and this is regulated through feedback inhibition from lowered glucose, which is the effect of insulin release from the pancreas beta cells. The brain switches from glucose to fatty acids in a state of starvation as ketone bodies and amino acids are catabolized, so you're losing protein in the most severe forms of starvation. The triacylglycerols, a normally sustained person can sustain that individual for 3 weeks, an obese person for a year. The epinephrine is a fight or flight hormone. It originates in the adrenal medulla. It dilates respiratory passages, increases heart rate, raises blood pressure, stimulates glycolysis, and inhibits insulin secretion. Cortisol is a hormone which responds to stress, is a slow-acting hormone. It releases fatty acids, protein breakdown, gluconeogenesis, and increases glycogen stores. Diabetes is a common disease. It's present in 9% of the U.S. population and 25% of the U.S. population over 65 years of age. There's type 1 and type 2. Type 1 is insulin-dependent diabetes. It generally, but not always, has an early onset. This is judged to be caused by autoimmune destruction of pancreatic beta cells, and there's treatment by insulin. Type 2 diabetes is non-insulin-dependent diabetes. This is an older cohort. More obese adults fall into this category, although not exclusively. It's seen in children commonly. And these have minor symptoms, or milder symptoms, I should say, but no less serious, and they can culminate in significant morbidity and not insignificant mortality. Fatty acids become the principal energy source. Acetyl-CoA inefficiently is oxidized in the citric acid cycle because the high ratio of NADH to NAD inhibits the tricyclic tricarboxylic acid cycle. Ketone bodies are produced from increased acetyl-CoA, and this is produced in excess, and we call this state ketosis, and this creates acidosis or ketoacidosis, which is life-threatening. These patients oftentimes will have wild fluctuations of glucose, and the glucose can be into the mid to high three-figure concentration. We talk about body mass index, the BMI, which is the weight in kilograms divided by the height in meters squared. By and large, less than 25 is normal, 25 to 30 is overweight, over 30 is obese. And when excess calories are consumed, there are three options for them. They are converted to fat, burned by exercise, or converted to heat. The organism has, or at least the human body has various entities which direct and convey the state of satiety to the body. Among these are leptin. Leptin is an adipokine which feedback inhibits fat synthesis when satiety is reached. There are genetic deficiencies in lectin, and they create diabetic-like conditions. These act on the arcuate nucleus of the hypothalamus, and they decrease appetite. Neuropeptin Y stimulates appetite, opposed by alpha-melanotic stimulating hormone, which is alpha-MSH. Leptin stimulates a transmembrane domain Janus kinase, JAK, phosphorylation activation, and stimulates alpha-MSH. Leptin is often elevated in obese subjects, suggesting a defective receptor. So the concentration of leptin does not correlate to obesity. Adiponectin increases its insulin sensitivity. It stimulates AMP-activated protein kinase, AMPK, and this acts systematically. AMPK has a broad array of functions. It shifts metabolism towards energy generation. AMPK also is stimulated by exercise. It catabolizes glycolysis, beta oxidation, and glucose transport. It inhibits gluconeogenesis and fatty acid synthesis. AMPK also stimulates beta oxidation by phosphorylating and inactivating acetyl-CoA carboxylase. So when we look at this, we have a binding glycogen binding domain here, a beta subunit, an activator site. The alpha subunit, this is a catalytic domain here, and then there's a nucleotide binding domain here with nucleotide binding sites and the gamma subunit at this location. There's peroxisome proliferation, activated receptors, a family of ligands which activate genes utilized in lipid metabolism. These ligands vary in type and they are site-specific. Short-term appetite stimulators, ghrelin, is produced in the stomach, intense hunger sensation. The family of appetite reducing entities, which are widely popular and becoming more and more available, act on this pathway here with ghrelin. They help to inhibit this action and cause the body to experience satiety at the cost of muscle mass, which is a side effect when you're shutting down one part of the body energy stores. Also, endocannabinoids, they heighten the sensory response, which is produced in animals, and this accelerates increased consumption of foodstuffs. Also, there is the effect of microbiological gut flora. Probiotics disfavor adipogenesis, and prebiotics are nutrients which facilitate probiotic growth. In diabetes type 2, we talk about this being an epidemic. Epidemic is, by definition, an unexpected increase above baseline. This is a state of insulin resistance. Initially, it's a lack of sensitivity in muscle and liver, and the beta cells of the pancreas eventually fail. In the mechanism of action, lipocytes cannot store excess triacylglycerols. The lipocytes release proteins which incite a macrophage-mediated inflammatory response. The free fatty acids form lipocytes in various tissues, liver, muscle. Regulation is altered in these sites. The therapeutic drugs that are used, particularly in type 2 diabetes, target the ability of the body to convert fatty acids to triacylglycerol. There is a genetic component to this type 2, and not all obese people develop type 2 diabetes, but it is a characteristic that is seen obesity, but is not exclusive. So when we talk about treatment modalities, we have the time-tested weight loss, which is directed at adipose tissue, reducing the triacylglycerol content. This has the effect of reducing the lipid burden, increasing the capacity for lipid storage in adipose tissue, and restoring insulin sensitivity. Exercise acts on the AMPK, which is activated by increasing the ratio of AMP to ATP, and this aids in weight loss. Bariatric surgery has the effect of leading to weight loss and better control of blood glucose. This is a case where there is literal stapling and removal of the stomach, so the stomach has a smaller volume and satiety is reached more quickly. This is obviously accompanied by certain lifestyle changes. Sulfonylureas such as glipizide, glyburide, and glimepiride, beta cell blockers. They act on pancreatic beta cells and the potassium channels. They stimulate insulin secretion by pancreas, so these are used in type 2. There is big quantities of metformin, which is also activates AMPK, decreasing glucose uptake by muscle. Thiazolidine diones are used. These act on PPAR, which is PPAR is a peroxisome proliferator activated receptor, and these are nuclear transcription factors which are activated by lipidic ligands that alter the expression of specific genes. That will act on the liver, muscle, adipose tissue, increase glucose uptake. And then GLP-1 modulators are glucagon-like peptides, and they enhance insulin secretion by pancreas. And that's it for this lecture. Thank you very much.